Okay, so welcome back friends. Welcome back in another video tutorial from microbiology with Charlie. So in this video tutorial we are going to discuss about a new topic of recombinant DNA technology, that is the molecular markers. Okay, so in this video lecture series we are going to discuss about RAPD, RFLP, SSR, SNPD. Okay, so this video is just an introductory video of molecular markers. Okay, so let's get started. So I think if I told you to write a difference between a dog and a human, or let's say if I told you to write a difference between a monkey and human, so I think you can easily write, right? But if I told you to distinguish among you and your friend, okay, on the basis of morphology, okay, so you cannot write anything, right? You can just only write about that you look like you are tall rather than your friend, or you are healthy as compared to your friend, right? But you cannot derive the detailed difference between you and your friend, right? So for distinguishing between two human or between two organisms who are in same species, we are using molecular markers. Okay, so these molecular markers are used for variety of purposes, for example, let's say you are running a industry. This industry is of tomatoes, okay, and now you have to identify that which tomato is more tasty and whose tomatoes demand is high in market, right? So you can distinguish them with the help of molecular markers, and now you will only produce that particular type of tomato, so for your profit, right? Another important thing, that there are various kind of paternity problems. For such kind of thing, those molecular markers can help you to determine the biological father and mother of a particular child with the help of DNA fingerprinting. Remember it very carefully that we are using molecular markers for DNA fingerprinting also, right? So now it is necessary to determine, or it is necessary to understand what is molecular marker. So a molecular marker is nothing but a sample. Okay, that sample can be DNA, that sample can be RNA, that sample can be protein, or in sometimes that sample can be a isoenzyme, which is a biochemical. I hope you know. Okay, so under such circumstances, what is the utility of these samples? So this sample can be easily isolated from both the individuals, okay, and not only that, you can easily test them in laboratory in minimal cost. Okay, that means molecular markers are what are molecular markers? So molecular markers, or any simply markers, are nothing but samples, okay, that can be easily isolated and tested in laboratory in low cost, and they help us to determine the differences between two individuals of the same species. Okay, so this is the technical definition of the molecular marker. That is, molecular marker is defined as any DNA sequence, okay, that can show polymorphism. Okay, so polymorphism is what? Polymorphism is nothing but the presence of genetic variation in population, okay, upon which natural selection can operate and can be detected using a molecular technique, right? That means here we are using genetical molecular marker, or simply genetical elements to distinguish between two individuals. Okay, so let me tell you that in humans there are 99.9% similarities. Okay, only the 0.1% difference is detected by molecular markers or any kind of markers that we are using. Okay, for example, DNA markers or RNA markers or isoenzyme that we are using, I will going to discuss about it. So among them, the most important are the DNA markers. Okay, so what is molecular markers? I think now it is clear to you that molecular markers will help you to determine or to distinguish between two organisms of same species. Okay, that can be human, that can be any organism. Okay, and with the help of this you are simply isolating a sample from both the organism and then you are testing it in the laboratory, okay, to find out the distinguish between the two organisms on the basis of the sample you have isolated. Oh, okay, for example, let's say from you I have isolated a DNA sequence, okay, and also from your friend I have isolated a DNA sequence, and let's consider it is a molecular marker. Okay, so now this molecular marker will help me to distinguish between you and your friend. Okay, that is the idea of molecular marker. So this molecular marker is mainly DNA sequence is in most of the cases, and it is a DNA sequence which can be of many base pair. Okay, mainly it is 10 to 15 nucleotide, or it can be a small change in one base pair, or you can have a repeating sequence as a molecular marker also. Okay, that means more or less you can say that it is a DNA sequence. Okay, although we are using RNA also, but DNA is important for us, so DNA. Okay, now so molecular markers are generally divided into two parts. One is genetic markers, another one is biochemical markers. So among genetic markers, DNA is mostly useful. Okay, we are also using RNA markers, and in biochemical markers we are using isoenzymes. Okay, so the idea is we will isolate them, we will run them in polyacrylamide gel if we are using protein, or in agarose gel simply if we are using DNA as a molecular marker. Okay, now I have also told you that it can be a very large sequence, or it can be a 10 to 15 nucleotide sequence, or it can be a change in one base pair, or repeating sequence, or can be a single base. Okay, so now there are two important characteristics of a marker that you have to keep in your mind, that the DNA sequence that you have chosen for using as a molecular marker should be easily quantified and detected. Okay, that means detection and quantification of the marker should be done at a low cost, because I think you can understand that here we are using those molecular markers as industrial purposes, right? And there is a basic funda of any industrial approaches that we will invest less money but we will get more profit from it, right? So we will have to choose only those kind of DNA sequence that will give us these properties, that it should be easily quantified and detected. Okay, so now whenever we are using isoenzymes, we can detect them with the help of chemiluminescence or fluorescence. Okay, and the fluorescence technique can also be used in case of DNA also. Okay, where we are using hybridization. We have already discussed about hybridization in our colony PCR or any other kind of video that we have discussed until now, right? Now there is an interesting thing that I want to discuss, that is molecular markers on the basis of genetics are divided into two types. One is dominant marker, another one is codominant marker. Before that, it is necessary to understand about the basic principle of Mendelian genetics. Okay, so in Mendelian genetics there are two things, that is dominant and recessive. Okay, so I hope you know about what is allele. So allele is the two different form of a same gene. Okay, so now this capital letter is used for saying dominant type. Okay, dominant type or dominant allele. Okay, and the small one is used for referring recessive. Okay, so what is the funda of dominant and recessive? The idea is here we are using genotypes. So what is the genotype? So genotype is nothing but a symbol that we are using to showing the genotype of a particular organism. Okay, so here we are using only one property, that is the color. Okay, let's say for example, so we can use genotype of many properties also. For example, let's say TTRR. So here we are using two properties. One is the height, another one is the shape, right? So here I am only using the color. Okay, so among them, when you are using any capital letter here, this capital letter will help you to understand about the dominant type. So what is the meaning of the dominant type? The meaning of the dominant type is whenever this allele is present in the gene, make sure that the phenotype will be the phenotype generated by that capital B. Okay, for example, let's say this capital B is for blue color. Okay, so the phenotype of that particular genotype capital B small b will be blue. Okay, I hope you can understand. And whenever you will get something like that, you will not get the phenotype as blue. Okay, because here you can see both the two are recessive, right? So that is the idea of dominant and recessive. Whenever dominant is present, make it very clearly that the phenotype will be determined by the dominant allele. Okay, and whenever no dominant allele is present, make it very clear that the recessive the recessive allele will determine the phenotype. Okay, so now this is the three possible option. One is homozygous dominant. Okay, so I hope you can understand what will be the phenotype. The phenotype will be blue color, and again this is the small b. So I can expect that you can also understand about the what will be the color. So here blue color will be not observed, right? So simple white. And in case of heterozygous type, you will get, yes, exactly, blue color. Okay, so now we are using two kind of molecular markers on the basis of genetics, that is dominant marker and codominant marker. And remember, I have told you that molecular marker is nothing but a DNA sequence that is helping you to determine the difference between two individuals. Okay, so here the idea is, let's say first of all we will discuss about the dominant marker. Okay, so we have isolated the DNA from father, and we have isolated the DNA from mother, and we are isolated the DNA from offspring. And I want to told you that here we are solving a paternity problem. Okay, so now we will run the sample in agarose gel. Okay, so now after agarose gel running, when we are using the UV transilluminator to detect the band, we get band in here when in on the basis of the father sample, and we have get the band here on the basis of the mother sample, and here we have also get band, right? So now on the basis of this, it is very clear that the father sample simply indicating that the offspring is the biological son of that father. Okay, because you can see that the molecular weight of the molecular weight of the DNA is exactly similar with the DNA which is present in the offspring, right? But you cannot get any indication whether the mother is the biological mother of the offspring or not, right? Because you can see that the offspring samples do not giving any band that is equal to the band we have get in mother's lane, right? So now under such circumstances it is obvious that we can easily conclude that yes, the offspring is the biological son of that father, right? And we can also conclude that the genotype should be dominant, right? So now let's try to remember that I have told you that both homozygous genotype and heterozygous genotype are same in the terms of phenotype, right? Now let's try to think about this. Can this entire experiment can told you that with what genotype the offspring has? No, because it is only indicating that dominant gene is present, but it not indicating that whether there is a presence of recessive allele or not. Okay, so that means, remember it very carefully, that is the dominant marker can help you to distinguish between the dominant and recessive allele. Okay, because you can see that father's allele which is here dominant and mother's allele which is here recessive can be easily distinguished, right? But it cannot distinguish whether a particular genotype, let's say here in case of the offspring, the genotype is homozygous genotype or heterozygous genotype. Okay, so that is the problem of the dominant markers. Okay, we can use dominant markers to distinguish between distinguish between dominant genotypes and recessive genotypes. Okay, no problem, but we cannot distinguish between homozygous and heterozygous. Okay, so under such circumstances another type of marker technique is developed, that is codominant marker. Okay, so in codominant marker you will get two band that will give you the clear indication that yes, this offspring has this father as a biological parents and also this mother as a biological parents. Okay, and it also help you to determine the actual genetic makeup or genotype of that offspring, right? Because now you can easily write that the offspring is heterozygous, right? I hope there is no doubt about this, right? So the example of dominant marker is RAPD. On the other hand, the example of codominant marker is RFLP and SSR. Okay, we will going to discuss about each of the marker in details in upcoming video. So till now we have discussed about the molecular markers in a very basic level, that molecular markers are nothing but a DNA sequence, okay, that can show polymorphism. Okay, that means with the help of this DNA sequence you can distinguish between two individuals coming from the same species. Okay, so we have divided the molecular markers, I am mentioning it as MM, into two parts. One is genetic markers, another one is biochemical markers. We have taken isoenzyme as the biochemical markers and DNA, RNA as genetic markers. Okay, among them the DNA is mostly important, and in DNA we have discussed that the DNA sequence that we are using can be a very large sequence or can be a DNA sequence having difference in 10 to 15 nucleotide, or can be a single base pair change, or can be a repeating sequence, okay, or can be single base. Okay, and another important point, that marker should be easily detectable and quantified. And we have also discussed that molecular marker can be divided into two parts. One is dominant marker, another one is codominant marker. Okay, so dominant marker help you to distinguish between dominant allele and recessive allele very clearly. but it cannot help you to distinguish between homozygous and heterozygous. Okay, for doing this you have to switch on codominant markers. Okay, these codominant markers will help you to understand about the difference between homozygous and heterozygous, and also it will help you to distinguish the genotype of the offspring. Okay, so that is the idea of the molecular markers. So I hope this video will be helpful to you to understand the basic concept of the molecular markers. So we will again discuss another type of markers in subsequent videos. So till then, bye bye.